And if you talk about autoantibodies in immune regulation, then also immune evasion is a topic that's related to that. And Farzin Mashreghi from the DRFZ, Deputy Director of DRFZ, will now give his talk on immune evasion of SARS-CoV-2. And that is also something that viruses have really developed. Maybe we experience this online with a new virus in the population, that there might be adaptation processes, including evasion. And Farzin, we are interesting to hear what you tell us about this virus.
Yeah.
You change your title, doesn't matter.
Yeah, I changed my title, so because the topic. So first of all, thank you, Andreas, for inviting me to this cool meeting. And I was asked to give a talk about immune evasion, and the only pathway which I investigated during the pandemics was the TGF-beta mediated pathway. And this is actually, I do a recap of the last years. And this was the study which was done by predominantly Marta together with Andreas' group and also Fritz Melchers, who passed away, sadly. And we actually saw that it was very early during the beginning of the pandemic that plasmablasts are chronically visible in the peripheral blood of patients who develop severe COVID-19. And we decided to take a look at these cells, isolated them from the peripheral blood of severely diseased SARS-CoV-2 or severely diseased COVID-19 patients, and did high-dimensional single-cell transcriptomic and repertoire analysis of the cells. And what Marta could show is that when we look at these patients longitudinally in the beginning, when they're in the ICU, they show a plasma blast for predominantly predominantly showing antibodies again with the isotype IgG2, which indicates that they are performing a isotype switch due to high levels of interference. Then in the middle of the observation period, we saw that this plasma blast switched further to IgG1 and IgA1, which indicates the presence of IL-21 and TGF-beta in these patients. And the more patients stay in the ICU, we saw that the immune response is actually dominated by high levels of TGF-beta. And when we looked for this plasma blasts, which were expressing IgA2, they were not present in the lung tissue. And that's very important. They were not directed against spike and the M protein of the virus. And if you clone these antibodies or the B-cell receptors, which these plasma blasts express, most of them are directed against self-antigens, for example, against nuclear antigens. And this is very similar to diseases like lupus. And the question was whether this is a figure from Professor Iwasaki. This I am modified a little bit, that delayed interferon signals, which we heard can be about inborn errors of immunity or the development of autoantibodies, where the TGF-beta actually is also responsible, that in these people who get severely affected by the infection, the disease becomes severe. And we questioned whether high levels of TGF-beta or untimely high levels of TGF-beta can actually contribute to the disease. And this was the case. This was done together with Andreas Stiefenbach's group, together with Mario Witkowski and Caro Tizian. And what we could observe that if you look at flu-like diseases or ambulant COVID-19 patients, moderately affected COVID-19 patients, and severely affected, you can appreciate that the levels of TGF-beta are way too early. And you see high levels of these, significantly high levels of this cytokine within the serum of these patients which were hospitalized. So, and you can use this actually and transform it and use this 2 weeks after infection already, or before they were submitted to the ICU, that the levels of TGF-beta are actually indicative or predictive for later hospitalization and even for mortality. So how can SARS-CoV-2 actually trick the immune system? So there was us and other people showing that here from Munich, from Stukalov et al., that SARS-CoV-2, ORF8 and ORF3 can actually interact with the intracellular domains of the TGF-beta receptor. There is one study from Berkeley showing that actually spike interacting with the integrins on endothelial cells in a TGF-beta dependent manner can lead to vascular leakage, and most importantly, that the M protein of the SARS-CoV-2 can interact with the transcription factors which induce the signal transduction into the cells and can promote the signal transduction by it, which leads to more induction of TGF-beta transcription factor, and this is shown here, that if you look in single cell nuclei, that those cells which are actually infected, these are the red bars, they produce also more TGF-beta and TGF-beta dependent genes, which activates the TGF-beta, and this shows that actually SARS-CoV-2 infected cells can also produce more TGF-beta. So now to the more recent studies which we did, and this started when Thilo, who is also in the audience, called me and told me that they see a lot of children affected by a hyperinflammatory toxic shock syndrome, which is now called multisystem inflammatory syndrome in children. And this is very interesting that this hyperinflammation occurs 4 to 6 weeks and is associated with prior SARS-CoV-2 infection. So these children get infected, they develop a mild disease or even asymptomatic. And in 1 out of 4,000 children, this disease, so very rare, can develop. And this is described as a non-specific hyperinflammation, and the mortality is about 0.8%. They develop heart symptoms, scratches, brain involvement, and so on. So what we did, so they also developed a treatment for it, which is very good. This is corticosteroids and intravenous immunoglobulins, and this led that we, in Germany at least, there were not one single dead child. So what we did a systemic approach, analyzed up to 50 different cytokines, and what caught my interest is that we saw TGF-beta levels rising in those children which developed MIS-C compared to those which had infection but did not develop 4 to 6 weeks after the infection. High levels of TGF-beta, and if you give them corticosteroids, the TGF-beta drops again. And if you then compare it to elderlies, then you see that the TGF-beta levels are almost as high as those patients which suffered from severe COVID-19. And that was interesting because we had here a post-COVID sequel in children, and there is one mechanism which both diseases have in common. It's namely the high levels of TGF-beta, and this TGF-beta in the serum. So it's normally TGF-beta is in a unfunctional state. It has to be cleaved to be active, and this is shown here that the TGF-beta which we measured and which we see is actually active, and we see phosphosmut 3 phosphorylation, and you can block this very fine with blocking antibodies against TGF-beta. So the question was whether this active TGF-beta high levels in these children does anything to the cells of the immune system. So we did a little bit differently than others do. We went for, we heard already, for plasmablasts and activated T cells. So we isolated them according to CD838 and HLA-DR expression. We took the memory B cells out, some of the monocytes, and plasmablast from frozen material is really hard, but we tried also to capture them. So I don't want to bore you with single cell analysis. The key data in the monocytes is that one of the pathways which turned out to be highly significant is that those patients which develop MIS-C have reduced ability, or this is the prediction, to present antigen. And the second point is what we learned from our TGF-beta story in severe COVID is that the gene sets which are upregulated and associated with TGF-beta are upregulated as well in the T cells, in the activated T cells, which we analyze. So there is less antigen presentation. There is a TGF-beta signature in the activated T cells, which might compromise their reactivation. So we went on and wanted to functionally test whether indeed the T cells from MIS-C are unable to get reactivated towards antigens, and we used the method which was developed by Andreas Thiel and refined by Alex Shefford, also in the audience, that we took MIS-C blood samples from the acute disease and looked in the follow-up and re-stimulated the PBMCs in the presence of anti-CD28, anti-CD40, and different types of peptides, assuming that the children had some viral infections. So the peptide pools are depicted here. It's herpes, EBV, CMV, HHV6, adenovirus, and SARS-CoV-2. And I just want to make a long story short, is that for CD8 cells and CD4 cells, you see that during the acute phase, there is less, the memory T cells, they have less potential to get reactivated, and after once they go into remission, this reactivity is increased. So we did also serum transfers to show that the serum, the TGF-beta levels we are in the serum of these children are actually functional and able to modulate this, and did the same assay, so the RT assay or the T cell activation assay, and can show that if you do the serum transfer from MIS-C on healthy donors which used to have memory cells reactive to this pathogen-derived peptide pools, and you can see you can suppress the reactivation, but this suppression of reactivation can be actually reverted by blocking the TGF within the serum of these children. So there is another another key association of MIS-C, so it's actually used to diagnose MIS-C, is that the children show an expansion of CD4 and CD8 cells expressing the TCR-V beta 21.3. And so if you look at this picture, is that people who described it could even associate that the more expanded TCR-V beta 21.3 expanded T cells you see, the more severe is the disease. And we looked, because we had the repertoire, we looked in our activated T cells, and all of the children which we actually investigated showed increased levels of these T cells which express this beta chain. And I guess we were able to see this because we went for this activated T cell population, which we pre-sorted via CD38 and HLA-DR expression. And this was not only in the Berlin cohort, we collaborated with Alex Belot, who originally described this finding, and also with Helen Su from NIH with a cohort from South America, where we could show that indeed this beta chain is increased in the activated T cell compartment, suggesting that these T cells saw something which led, like we see it in vaccination. If you vaccinate a person 7 to 8 days later, you see this antigen reactive T cells appearing in the peripheral blood of the subjects. And this was not by chance. If you look for the V alpha expression, they really paired with certain alpha chains, and we tried also to mimic this and understand maybe by looking at their repertoires, at least of the V gene usage, we can identify what kind of reactivities these cells have, and we try to match the HLA best to the patient cohort which we had. So there were nothing significant, a little bit of increase of the HLA haplotypes which are indicated here, and took people with the same, almost the same HLA, and tried to build up a library of antigen reactivities with the RTSA, so with 5 people with EBV, 5 with CMV, 3 SARS-CoV-2, 3 people who were vaccinated with weasel or where we reactivated cells for measles, and with adenoviruses did the RTSA, got the antigen reactive cells due to CD154 expression and CD137 expression for the CD8 cells, and did a single cell sequencing of them combined with repertoire. And this is what you get when you look at this antigen reactive cells for all these pathogens or peptide tools from these pathogens. And then you can appreciate that you have a lot of antigen-specific cells via CD40 ligand expression. You have CD8 and CD4 cells. You have the cytotoxic cells. You have the follicular cells indicated by IL-21 or ICOS. We have interferon gamma expressing cells, and the important information is that all the 21.3 expressing cells, this is the gene TRV-B11.2 for the RNA, that they are almost marking the ones CD4 and CD8 cells, which are rather cytotoxic. And then we just compared the beta and alpha usage of the MIS-C patients with those which we generated in this library. It's around 20,000 cells. And what came out is that the MIS-C patients actually clustered together with 4 out of 5 EBV specific cells, antigen-specific cells, which we isolated and sequenced, showing that there might EBV reactivation might contribute to this hyperinflammatory disease. We went back to the clinics and looked for the serology. And what we did here is that we looked for those kids which did not get IVIG before. So to not influence that, then we had 62 from France and Berlin, which we could compare to the age match control group from Berlin-Brandenburg era. And as you can see, is that almost 80% of the children in the age, so the median of 6 to 8, had already contact. So this is highly significant with EBV before. And if you go for those children which had SARS-CoV-2 and did not develop also this, so that they are rather lower in seropositivity. And then we looked also at the levels of the antibodies in the serum of these kids. And as you can see here, so they have higher levels, for example, for EBNA1 here, showing that there might be some acute EBV reactivation ongoing for IgG, as well as for IgA1, also indicating the involvement of TGF-beta. So then the final proof is, do we see actually EBV? And unfortunately, we hadn't collected saliva from these kids. And then we thought we go and just sequence all the activated B cells and plasmablasts, which we get out of our cohort. And here it's depicted, left are the memory, right, always the penguin structure are the plasmablast. And we looked for EBV transcript here. This was the EBNA2 transcript, which you can see there in the UMAP. And this is actually shown here. It's very similar to severe COVID. So there we also see, as a control group, as a positive control group, plasmablast or memory B cells, which show transcripts for EBV. So actively EBNA2 expressing cells, which indicates the latency 3 of the EBV cycle. And as controls, So to mimic acute inflammation, we took people who got the first shot of an mRNA vaccine or the AstraZeneca vaccine. And as you can see, after primary vaccination, at least we do not see EBV reactivation. And so what is the epitope which this T cells react to? And we were really lucky. And so this paper in a Journal of Immunology, where they actually investigated one single case of EBV infection and took for CD4 cells a tetramer with a specific peptide, which is marked there from EBNA2. And as you can appreciate, so the TCRV, can I, you don't see this, or? It's digital. Okay, so you see that the V beta 21.3 tetramer bond, this peptide bond to this tetramer is actually able to mark TCRs expressing this specific beta chain, which we see also in MIS-C. And we ordered the peptides, a shorter form and a longer form, and did the T cell activation assay for CD4 and CD8 cells. And as you can appreciate, the antigen reactive cells have higher expression of 21.3 expressions, showing that they are actually reactive to EBNA2. So we did also these predictions, whether it's HLA1, HLA2 tools, and funnily, the predictions which are available, which we also saw today, told us actually that there are very weak binders to both. Yeah, so we are very surprised to see this, but happy because it's highly significant. And we could show that actually these T cells react to EBNA2-derived immunodominant peptide. And then the final proof was, are these 21.3 positive cells actually able to kill EBV transfected cells? And we did LCLs. We heard about this also today. So we immortalized B cells with EBV and co-cultured them in an autologous fashion, depleted rather the V beta 21.3 cells or enriched for them, put them together, and looked whether there are better killers of EBV infected cells. And this is shown here for CD8 cells in a 1-to-1 ratio. This is indeed the case for 7 donors. And they are also, once they come together, they are also secreting, so they are cytotoxic and express CD107, a marker for active secretion. For these CD4 cells, we had to go for 30-to-1 because only a fraction of the CD4 cells were really cytotoxic, I guess, but a similar picture. And TGF is actually known to suppress the cytotoxicity of T cells. So this is the conclusion. So for the MIS-C, we think that kids who have encountered already EBV, maybe some genetic factors, which Professor Casanova already showed for us, 1%, I guess, of the children which suffer from inborn errors of the type 1 interferon pathway. And we think that then we get a SARS-CoV-2 infection. This we know can trigger in these children within 4 to 6 weeks. It might be that this virus is somewhere or spikes somewhere present in these children. This induces a TGF-beta induction and leads to dysfunctional T cells, which are not anymore capable to keep this latent viruses. For, by the way, also CMV was one of the reactivities which we saw to be significant, and this EBV induction, uncontrolled activation, we think that this also participate to the hyperinflammation. Okay. And these are the people who were involved. So it was a lot of people from Italy, from the US, from Chile, here from the Charité, the pediatrics. The first author, Götze Tillmann, who actually was very busy getting the samples and seeing the patients. Mario from Andreas Diefenbach's group, from the BCRT who helped us out with the LCLs and killing assays, and Mona and Pavel from my group, which helped us to analyze the sequencing data. Thank you very much.
Thank you, Farzin, for this amazing link between TGF-beta signaling and also the EBV reactivation. That's, by the way, frequently seen from long COVID patients. That will be my question. Carsten.
Did you have a chance to look at the NK cells, because they can also target EBV transformed B cells, and it would fit with your TGF-beta story again?
So this is what always Mario says. Why did you not sort for NK cells? We didn't look at it.
Next question.
So there's this is a very, very nice story. So there's different EBV viruses, different lines of viruses. Did you have a chance to look in your children?
So this peptide is derived from type 1 EBV viruses. It's completely missing in type 2. And very conserved, actually.
So then you use B95.8 as virus strain to
the standard one, but we ordered now different strains to test.
Because that would be also my question, whether that could be a reason why some of individuals are reactivating their EBV responses, or EBV is reactivated and others not, and that might be also a difference.
So EBNA2 is always used for differentiating between type 1 and type 2. That's true.
Yeah. My question, whether you mentioned briefly CMV. So do you think that it would make sense to look at the long COVID individuals, because you had the MSC the pediatric cohort, but there is a finding that many studies have shown that there are always subgroups of long COVID individuals with reactivation of EBV.
One can look at actually the Chiara Romagnani NK cells, whether they pop up.
Also on top of that, and whether they're regulated by TGF-beta or negatively regulated.
I think so, because it also compromises the activity of the NK cells. So.
Last short question. While Live gets the microphone, how do the CD4 T cells kill?
With granulysin, perforin, which they express, actually.
Live.
Yeah, super story. Did you measure viral titers in some compartment in these kids? So there must be lots of cohorts with samples that.
So we had to do that because Review asked, so we went worldwide to get actually saliva, because in the serum or plasma, it's very hard, it's very unsensitive to get them. So what we could in half of them see indeed with PCR EBV, but only in a fraction where this was done actually. So we had problems with material. So the gold standard would have been saliva, which we completely missed. Therefore, we went and isolated in B cells. So very expensive experiment, because normally if you want to see an EBV infected B cells, you have to sequence about 1 million cells in a healthy person. And here it was actually 1 transcript in 300 in severe COVID-19, as well as in MSC patients. So it was the best way to show actually to get the cell which is now actually producing the virus. It's activated plasmablast.
Okay, because in clinical settings you do detect it in blood and in severely ill patients. That's how we do it. And there's also some manuscripts out there, I don't think they have appeared yet, that measured a whole bunch of, chronic viruses and the virome or whatever, that obviously not only EBV, but other viruses that get reactivated in different, let's say, COVID-associated conditions, both in the severe stage as well as in those that go on to develop sequelae.
It's actually true also for HIV and so on.
Yeah, and that again shows that this mechanism might be relevant for a general loss of immunocompetence, and that is then leading to a long progress of other infections that are then feeling that they could come up now or never. Super, thank you so much.